Hello everyone, welcome to the Study Companion. This is Tushar Biswas and we today are going to study the DNA. The DNA double helix. It's a basic introduction to DNA and I hope we'll all like it, we'll learn from it, we'll grasp it, the lesson properly. Without further ado, let's begin. Let's begin. Let's see now. DNA. DNA, the full form of DNA is deoxy. Full form of DNA is deoxy ribo. N stands for nucleic. A stands for acid. Now why D? Because generally whenever we come across D in bio, it stands for di, right? But this time it's standing for D. Like why D? Why D? We are coming to it a bit later. Okay, this is not di. This is D. Why D? The parts of the DNA. Parts of the DNA is number one, a pentose sugar. Number two, nitrogenous base. Number three is a phosphate group. And this pentose, we all know organic chemistry by now, and for people who don't know, there will be a dedicated lecture on not lecture, dedicated class on organic chemistry later on in this channel. Of course, pent means five carbon. Pent five carbon. Okay, now discussing the structure of the DNA. Discussing the structure of the DNA. Let us see what do we have in our store. Okay, parts of the DNA. Parts of a DNA. Parts of the DNA. Remember these. Don't copy this one down right now. Pentose sugar. Pentose sugar. Generally, a pentose sugar looks like this. Pentose sugar looks like this. 1 prime, 2 prime, 3 prime, 4 prime, and C over here 5 prime. OH over here. H, OH, H, OH, and H over here. This is a normal pentose sugar. Normal pentose sugar found in RNA. But there's one very small piece of information that's pretty important. Each and every OH group is a reactive group. OH group C over here is H, OH, this. These groups are pretty reactive in nature. These groups are pretty reactive in nature. Like how much reactive? It's pretty reactive. Now see, this is normal RNA. Now going for DNA part, DNA of this same thing will look the same. Just there will be a small point of difference. There is a small difference, very small difference. 1 prime H OH, 2 prime the difference occurs over here. Wait, I'll show it. 3 prime H OH. First let me show the similarities. H and over here C 5 prime H OH. Over here this H is there properly. Now this time to reduce the stability, sorry, my mistake, to reduce the reactivity of this, they have removed this O and made it H. This H, this H is a non-reactive group and hence 2 prime carbon is now muted completely. 2 prime carbon will not function properly. Let's not say it won't function. Let's not say it won't function properly. It would function. Normal function it has, but it won't go under, and it won't undergo any kind of reaction as such, because the reactive group is done away with. They have lost OH in the process, is done away with. So this confers DNA. This is the DNA. The name of this sugar is known as 2-deoxyribose pentose sugar. Sugar is for people to know, actually, this 2-deoxyribose pentose. So a pentose sugar, instead of this, we will write this over here. 2-deoxyribose pentose sugar with a nitrogenous base and phosphate form the parts of the DNA. This, instead of writing the name over here, don't write this name, erase this one down and write this one. This is the one. Okay, this is the one. Now, this has been done away with. This has reduced the reactivity reduced, stability increased. Of course, anything that this DNA is our carrier of is the carrier of genetic material, carrier genetic information. This is like a CD drive, CD-ROM that carries information. Now this CD-ROM can be accessed to three ports. These two ports being this one was a separate port, none of the common to each. And this one, for example, was an HDMI port. For example, if this port was an HDMI port, these ports are the DVD CD ports. Okay, this one had a separate connection, separate work, so this one was required. This one, for example, this is a disc. This is a CD player, for example. This is a computer. Let's consider this one as a computer. It is a HDMI port. This is required right now, because whenever we want to connect it through HDMI and stuff, we'll require this. But over here there are two DVD CD parts. Hence, if like we can take any disk, any CD-ROM, place it on any part, and it will get connected. It will get connected. When we are in networking, when we are like forming a when we're connecting the computers together, for example, when we're connecting the computers together, we have a habit. We will, like, for example, there are some people who are connecting the computers together. Everyone, how are they connecting? All the 3 prime OH are being connected to the 5 prime ones. 3 5 3 5 3 5 3 5. But there was a mistake. There was a slight mistake. Someone made a slight blunder and then connected one 5 prime OH to a 2 prime OH, 2 prime CD disk. So there won't be any error, but the information that was passing from 3 5 3 5 3 5 now has gone very far and gone through 2 now. See, it was 5 3, then again, sorry, 3 5 3 5. Now this, instead of going like this, it has gone in this method 2. Now it's going like 2 5 2 5 2 5. See, it should have gone like this. Now there will be a slight loss in the information. Then it would there would be a slight loss. For example, if this is the vertebral column, consider that as a vertebral column. Why did I consider that? I'll tell you a bit later on, because it's not sorry, not a bit later on. Let's tell it. If we, like, we all know that this is known as a sugar phosphate backbone. This is a sugar. Then there comes sugar phosphate backbone. Okay, so this is the normal parts of it is sugar, nitrogenous base, NB, and this is the phosphate, right? Sugar, nitrogenous base, and phosphate. This is the sugar phosphate backbone. Sugar, phosphate, backbone. Sugar, phosphate, backbone. This part. Now this runs like this. Runs, runs like this. If I'm drawing a strand like this. Okay, okay, done. 5 prime, 3 prime, 5 prime, 3 prime, 5 prime, 3 prime. See, it's going starting from 5, ending at 3. Done. Now, had there been two available, we could have had seen 5 prime is going normal. 5 prime, 3 prime, then again 5 prime, 3 prime. Suddenly a jolt, suddenly a jolt, 5 prime, then it's going like this. 2 prime, 5 prime, again coming down. See this jolt over here, this small jerk over here, it has changed the shape. It should have been like this. Now it's like this. Information retained is more over here than here. See, because of this thing, we have lost the stability. Okay, this is more reactive, less stable. Okay, more reactive, less stable. See, it's reactive. 2 prime is also reactive, 3 prime is also reactive. Any of the phosphate can join with either 3 or either 2. Phosphate has a choice. Now it can join anywhere. It can join anywhere based on the joining of phosphate with 3 prime or 2 prime. The information retained in these will get altered. Hmm? The information retained based on the joining of this phosphate always phosphate group. Speaking of the phosphate group now, speaking of the phosphate group, I'll come to the nitrogenous base a bit later on. Speaking of the phosphate group, what do we see in the phosphate group? Phosphate joins with the 5 prime carbon, let this one be 5 prime, 5 prime carbon. Phosphate joins with 5 prime carbon. Then comes the sugar, whole sugar like this. Then again the 3 prime carbon joins with the phosphate. Again this phosphate joins with the 5 prime carbon. This one again forms a sugar. This is connected to it, not forms a sugar. Again the 3 prime joins with the phosphate. This is the structure. This is the structure. Okay, this is the structure. Had there been 2 prime available, the phosphate could have had also joined over here. This could have had happened. This could have had happened. This phosphate can join over here now. Now see, the things have turned around. The tables have, look how the tables have turned. This thing now starts from here. Now whole thing will start from here. 5 prime. Then again 3 prime, for example, now this time joins with 3. See, it has option. It might not always lead like this. It might also lead like this. Instead of this, we might get something like this. Sometimes this, sometimes this, sometimes this. See the loss in information we are getting. See, the areas over here and over here are losing on information. They are losing on information. See, the areas have been muted, the areas have been shortened, hence they are losing on information. So that this shouldn't occur, to prevent this from occurring, our DNA changed itself and removed this O, gave us deoxy. Okay, I hope everything is clear. This might always, like, there might be 2 prime again the phosphate decides to shift in the next part. This phosphate is in 3 prime, it can also shift to 2. The 2 prime phosphate might have come to 3. There will be loss of information because the chemicals energy binding and everything will be different in different parts, so there will be loss in information. Hence DNA was required to be the information hub for the multicellulars like us, because we have differential areas in our body. Now speaking of the nucleotides, Speaking of the nucleotides, nucleotides, uh, nucleotides or nitrogenous bases are the better term. Okay, no, nitrogenous base, not nucleotide. My mistake. Sorry, nitrogenous base or N2 base or NB. You can also write. These are of two types: purine or purine, whatever you like to say, and pyrimidine. Purine or purine consists of adenine or adenine and guanine or guanine. Pyrimidine, on the other hand, consists of cytosine or cytosine and thymine. Thymine or uracil. Okay, now their abbreviations will be A, G, C, T, and U. Thing to be noted. Thing to be noted. Over here, over here, over here. Focus over here. These three are common to RNA and DNA. These three are common to RNA, DNA. Only thymine is found in DNA, uracil in RNA, and nothing else. Okay, instead of thymine, if you see uracil present, it has to be an RNA. Instead of uracil, if you see thymine present, it has to be a DNA. Okay, I hope I'm making myself clear. In case I'm not clear to you, please let me know in the comment section. Okay, this is the thing. This is the thing. This is the idea. This is the idea. Now this is the base. I'll have one or two videos based on the introduction on this topic. I'll shoot one or two videos like that, and then the introduction is gonna be a pretty long because look at the topic, man. This is DNA after all. This is DNA after all. Guys, one more piece of information. Always remember this. Purines are always connecting with pyrimidines. For example, example of like the ones in purines are purines consist of adenosine and exactly guanine. Pyrimidines consist of cytosine and thymine or or uracil. Okay, so over here, adenine always binds to thymine, cytosine binds to guanine in DNA, but in RNA, adenine binds to uracil and cytosine binds to guanine as usual. Now, why didn't I draw any bonds over here? Let's check it out. Adenine, thymine have two hydrogen bonds in between. Cytosine, guanine have three. Why two, why three? When I'll discuss the structures of these individually, then I'll let you know why. Then you'll get to know actually like why two and why three. Similarly over here, ANU2, CNG3. Now to be more specific, it's like this: hydrogen, 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 H, 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 H and H, H and H. It's like this. Now these hydrogen bonds, I'm going to destroy the impression on you. These hydrogen bonds, HH bonds are weak in nature. They are weak. They are so weak, they can be summed up by weak van der Waals forces. They have weak van der Waals forces in between them. HH bonds are so weak that they have weak van der Waals forces in between them. Means a slight amount of heat, and these separate. Slight heating. H, of course, not the heat that is similar to the body heat, like 37 to 40. No, 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 no. 37 to 40. No, or else we will lose out on so much of information we have. Genetic information I'm talking about. H and H. Slight heat. Heat of Anything greater than 90 degrees Celsius? Not even enough to boil the water. 90 degrees Celsius, not even enough to boil the water, and it's separating the DNA. See, 90 degrees Celsius, and these part their own ways. Nice. This go its own ways, this go its own way. So that's out over here. You can copy this one down. This is pretty important. Any temperature greater than 90 is enough to break these bonds, to break the bonds, hydrogen bonds. So adenine thymine, for example, if heated heated under uh, heated under a temperature of 90 degrees Celsius, any temperature greater than 90 degrees Celsius, 90 degrees Celsius, 95, these bonds will break. Broken, everything is broken. Not broken that term. Broken. Now people are thinking about broken hearts and stuff. Come on, man. This is class 12. Focus on your studies. Focus on your studies. See, I can see the eyes glowing. I can see the eyes glowing. Come on. I can focus on your studies now. It's a time to study. What? Yeah, people are getting hearts broken. Okay. Focus on bio. Focus on bio or any other subject you want, you like. Of course, everything will fall back in place once your studies are back in place. So, hashtag motivation. Okay, so this thing now, let us discuss. You have seen this sugar, 5 prime carbon, phosphate, phosphate. Okay, now how does this phosphate attach? How does this phosphate attach? It attaches itself like this. It attaches itself like this. This C has H and H over here and OH over here. Phosphate comes over here. Phosphate comes. Phosphate comes. Takes takes away the OH, removes the OH and joins itself over here. That's how phosphate occurs. Same joining over here. This OH is over here. Phosphate comes to its position and replaces it. Replaces the H. Okay, that's how phosphate joins. Now this phosphate has joined. Phosphate has joined. Let's draw the molecular diagram. 5 prime C bonded with P. Now this is again a P which is bonded to a 5 prime C, which is again a part of a deoxyribose pentose sugar. Again P, see 3 prime. So it's going up like this, so on and so forth. Over here again. Now this OH you have over here H and OH, 1 prime end. At the 1 prime end you also have stuffs like this. Now I've said in a bit earlier, I said a bit earlier that this OH, each and every OH is a what do you say, is a reactive group, is pretty reactive. Okay, now let's see what does this OH do. Why did I explain it as an HDMI port, something other than the 2 prime and 3 prime OH? Why is it required? Why not removed? If OH at 2 prime can be removed, why not this? 2 prime was was a nuisance. Okay, 2 prime was a nuisance because it would do the same function as 3 prime, but just the conformation would be changed. Conformation means structure would be changed. I purposely removed this H because once I remove the OH, I can as well as it write like this. This thing can be also written as this, like this. Now this H binds to an N which is already bonded to a nitrogenous base. Again, this H can be written like that, H, which is again binding to N, which is bonded to a nitrogenous base. This nitrogenous base, I'm showing like for example, this is adenine, this is guanine. Okay, this has 2 bonds. Adenine will always bind to thymine, So one more nitrogenous base. This nitrogenous base is what? Thymine, exactly. This is again bound to 1' carbon, 1' sugar, but this time, mind you, this time the sugar is inverted. Sugar is inverted. Phosphate over here and 3' over here. Sugar is inverted. Of course, N over here, H over here. Again, this P bonded to the 3' end, which is again bonded to the 5' part. 5', this one is 5'. This is oxygen. Sorry, no, mistake. And there, this P is again bound to this 3' guy now, which is again upside down. It's bound to 3'. This is the H of 3'. This is the H of 3'. See, see. And this sums up the antipolar nature of DNA. 5' up, 3' down. 5' phosphorus is on top. It's beginning the chain. It's like this. It's beginning the chain. This is a highly magnified view, highly detailed view. We zoomed into this part. We zoomed into this part. Now we are going back. We are going back. 5', 5', 5' P over here. Then all the way goes down to the 3' P. 3' P is the forming the end. So this is the 3' P. Justified? Justified? Justified, nah? Come on, tell me. Tell me in the comment section. Come on, man. Justified. And this is the 3' P. So we can say it's starting from 3'. We can say that. It's all the way running to 5'. See? 5' carbon present over here. 5' carbon present over here. Which is binding to this P. Right? Binding to P. Hence we can say this is the 5' P. So see, it's anti-parallel now. This is these are parallel lines. So these will be anti-parallel lines. Parallel but opposite to each other, anti-parallels. See, now this nitrogenous base, this one has to be drawn a bit downwards, a bit more downwards. Okay, this one has to be drawn a bit more downwards. Oh yeah. Now let's see. This is the P bonded to a 3' OH. Then again, this is the 5' part. 5' part is a 5' carbon, which is again bound to the P. This is a 5' carbon over here, 5' carbon over here, bound to P. I purposely drew it like this. I like it isn't required. Let's not do it. See, 5'. See, 5'. Now this thing is in alignment. Yeah, this is O. Done. Now this H again, dot 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 N, bound to nitrogenous base. For example, guanine, which is again triple bound to cytosine. Triple bound to cytosine. Cytosine. Now this has a nitrogenous base, nitrogen again. Now this is hydrogen, which is bound to this. See, 5' to 3'. Now this P again forms. This is a 3' P. This is a 5' P. See, this is the anti-parallelism. Okay, this is the anti-parallelism, and that's all for this lecture. That's all for this lecture. Thank you for being a wonderful audience, wonderful students you are indeed. Thank you. Please keep studying, keep a follow up on DNA. I'll be posting new videos. I'm trying to post it every day because 12's exams are closer than they appear. Okay, because you will have your pre-boards and boards and everything whatnot. Like these last months of the class 12, the last memories you don't even get to make properly, because number one, the school closes, and number two is that due to the ongoing pandemic, you don't even have any kind of memories of class 12. Last memory was probably class 10 or 9. I don't feel to hurt you. I don't mean to hurt you actually. Okay, so thank you for watching this video. I'm trying to build up a playlist. Where the videos will be in 1 day gap of each other. So every day I'll in short to be short, I'll be posting videos on these topics: biotech for biology class 12, and the guys of class 10 ICSE board who wants to focus on class 10 parts, they can even watch this. Okay, that will be a bit of extra information, and of course, who doesn't want extra information? Showing off. So, speaking of which, thank you. Have a good day. Keep studying. If you like the content, please do follow. It's your own discretion. Okay. Keep a follow up. I'll help you out with your class 12 bio part. Okay. This is the study companion, Tushar Biswas, signing off. Thank you. One more thing, guys. One more thing will be that I'll be bringing short topics on DNA or short topics on biotech. Okay, there will be hardly 20 minutes, 30 minutes video, nothing more than that, because it's the last. It's nearly the end of class 12, and we need to focus on the quantity. We need to focus on quality as well as quantity. Okay, none of them one-handed, single-handedly won't work. So we need to focus on both. We need to balance them both. Quality you'll get over here. I can assure you that you'll get quality over here. Of course, quantity, same thing, you'll get it over here. You will get it over here. Now, each and every student have their own uh, preferences. It's for you to know. It's for you to decide. I'll be there. The videos will be in a rather shorter format, short parts of the whole syllabus, and you can just increase the speed 2x to 2x, and then you can just watch it right away in a zoom. Okay, 10 minutes quick revision for this. In case you want a 10-minute quick revision, just click to 2x and the speed increases. This is hardly 20 minutes or 30 minutes video. 10 to 15 minutes will you will complete this video in 10 to 15 minutes. Okay, thank you. Keep studying. This is the study companion Tushar Biswas signing off.